Hello, this is Peter Germain from the Advanced Biophotonics Lab at UMass Lowell. In this talk, I will discuss quantitative fluorescence polarization imaging for thyroid cancer detection. Studies show that most adults will develop one or more benign thyroid nodules during their lifetime. The American Cancer Society estimates increasing incidence of thyroid cancer, with about 44,000 new cases diagnosed annually in the United States. The standard of care for detecting thyroid cancer is fine needle aspiration cytology. This approach is labor-intensive, time-consuming, and expensive. It requires subjective visual assessments of cytomorphology and depends on the skill and experience of the cytopathologist. The Bethesda system for reporting thyroid cytopathology is used by pathologists to categorize thyroid cytology specimens as cancerous versus non-cancerous. Category 1 is non-diagnostic or unsatisfactory specimens. Category 2 represents the benign lesions. Categories 3, 4, and 5 are cytologically indeterminate categories, including atypia or follicular lesion of undetermined significance, follicular neoplasm or suspicious for follicular neoplasm, or suspicious for malignancy. Category 6 represents the malignant lesions. Overall diagnostic accuracy of the Bethesda system is low, approximately 60.2%, when including lesions from within the indeterminate categories. Our group is exploring thyroid cancer detection at the cellular level using fluorescence polarization imaging of methylene blue. Methylene blue is a positively charged phenothiazine dye approved by the United States FDA for human use. This method provides quantitative results in real time without tissue removal required at moderate cost and can achieve diagnosis in single cells. Fluorescence polarization imaging characterizes polarization of the fluorescent light with respect to the polarization of the incident light. It depends on the rotational diffusion of the fluorophore during the lifetime of the excited state. Fluorescence polarization imaging is sensitive to the local environment of the fluorophore, its binding, and fluorescence lifetime. Binding and viscosity restrict rotational motion, while fluorescence lifetime determines the amount of time available for altering fluorescence polarization. During the lifetime of the excited state, the fluorophore molecule is subject to rotational diffusion. This diffusion is more rapid for the unbound molecules, whereas binding to a large complex such as the mitochondrion of the cell, restricts rotation of the dipole. As a result, fluorescence polarization of the unbound fluorophore will be lower than that of the bound one. Similarly, fluorescence polarization will be higher for the fluorophore in the more viscous environment, which restricts rotational diffusion. Previous studies in human breast cancer and brain cancer cell lines demonstrated that methylene blue fluorescence polarization is elevated in malignant cells relative to normal cells due to greater uptake of positively charged methylene blue dye in the negatively charged mitochondria of cancer cells. Many cancers, including thyroid carcinoma, are characterized by increased mitochondrial membrane potential. Therefore, elevated methylene blue fluorescence polarization can be anticipated from thyroid cancer cells as compared to normal. For this study, we used an in-house built multimodal confocal microscope to acquire the data. The methylene blue was excited at 642 nanometers, and the fluorescence emissions were registered between 670 to 710 nanometers. There were 3 detection channels, including reflectance, copolarized fluorescence emission, and cross-polarized fluorescence emission. The system provided high-resolution images displaying cytomorphology, as well as quantitative images that revealed fluorescence polarization signals from single cells. The field of view was 205 microns squared using the 63x NA1.4 oil immersion objective. Axial resolution was 3 microns, and lateral resolution was better than 0.9 micron. The scanning rate was set to 9 frames per second. To investigate methylene blue fluorescence polarization of thyroid cells, more than 30 clinical samples containing over 3,500 cells were obtained from pathologically diverse, discarded thyroid tissue of 21 subjects. The clinical specimens included 13 samples from malignant tumors, including 1 medullary thyroid carcinoma, 9 papillary thyroid carcinoma, and 3 follicular thyroid carcinoma, 7 samples from benign nodules, including 4 multinodular goiter and 3 follicular thyroid adenoma, and 12 samples from normal thyroid tissues. The cells were scraped from freshly excised thyroid nodules or adjacent normal thyroid tissue, deposited in L15 growth medium, and transported to the Advanced Biophotonics Lab for imaging. The cells were plated in glass-bottom petri dishes coated with 0.01% poly-L-lysine solution to enhance cell attachment. Erythrocytes were lysed, then cell layers were stained with 0.05 mg per milliliter aqueous methylene blue solution for 20 minutes. The excess dye was removed by washing the cell monolayers 3 times with phosphate-buffered saline solution. Optical images of the cells were acquired, processed, and compared to the findings of clinical histopathology. The results were statistically analyzed using a linear mixed effects model. This slide shows example images of malignant and benign cells. The top row shows results for a Bethesda system Category 6 medullary thyroid carcinoma case. The bottom row shows results for a Category 2 multinodular goiter case. The fluorescence emission images display cytomorphological features of the cells. The medullary thyroid carcinoma cells are organized in loose clusters of cancer cells with eccentric nuclei, whereas the multinodular goiter cells are large and have abundant cytoplasm. Fluorescence polarization images provide a quantitative measurement for each cell. The scatter plots show the fluorescence polarization value versus cell size for all the cells in each sample. The red horizontal line at 0.245 represents the maximum cellular fluorescence polarization value in all the non-cancerous samples. In this benign sample, there were no cells with fluorescence polarization higher than 0.228. It can be seen that cancer cells are clustered around higher fluorescence polarization values, whereas benign cells are clustered at lower fluorescence polarization values. The clinical diagnosis of each sample was confirmed by histopathology. Average fluorescence polarization of the malignant sample was 0.270 plus or minus 0.028, whereas that of the benign sample was 0.196 plus or minus 0.015. This slide presents example images of cytologically indeterminate cells. The top row shows results from a malignant papillary thyroid carcinoma case. whereas the bottom row shows results for a benign follicular thyroid adenoma case. Both samples were indeterminate category 3 in the Bethesda system. Fluorescence emission images display morphological features, whereas the pseudo-colored fluorescence polarization images provide distinct quantitative contrast between cancerous and non-cancerous cells. The scatter plots reveal elevated cellular level fluorescence polarization values in the malignant cells relative to the benign cells. The red horizontal line shows the maximum cellular fluorescence polarization value in non-cancerous samples. In this benign sample, there were no cells with fluorescence polarization higher than this value, 0.245. The clinical diagnosis of each sample was confirmed by histopathology. Average fluorescence polarization of the papillary carcinoma sample was 0.263 plus or minus 0.019. Whereas that of the follicular adenoma sample was 0.204 plus or minus 0.018. Samples were organized in 6 histological groups, including medullary thyroid carcinoma, papillary thyroid carcinoma, follicular thyroid carcinoma, follicular thyroid adenoma, multinodular goiter, and normal. Fluorescence polarization of methylene blue was significantly higher in malignant samples for all cancerous, non-cancerous sample comparisons. The single medullary carcinoma case was excluded from the statistical analysis of histological groups. Analysis of 12 cytologically indeterminate samples revealed that fluorescence polarization in the malignant nodules was significantly higher than in benign. The samples were organized into 3 diagnostic groups, including all malignant, all benign, and all normal samples. Methylene blue fluorescence polarization was significantly higher in the malignant group versus benign or normal groups. The scatter plot shows fluorescence polarization versus cell size for all cells analyzed. Red diamonds represent malignant cells, blue circles are benign cells, and green squares are normal cells. There is pronounced overlap of cancer cells at higher fluorescence polarization values, and similar overlap of non-cancerous cells at lower fluorescence polarization values. Please note, the black horizontal line reveals that there are no cells in benign or normal samples with fluorescence polarization greater than 0.245. In summary, we investigated confocal fluorescence emission and polarization imaging of methylene blue stained cancerous and non-cancerous thyroid cells. Significantly increased fluorescence polarization of methylene blue was measured in cancerous thyroid cells versus benign or normal. Methylene blue fluorescence polarization provided reliable and accurate differentiation of cytologically indeterminate thyroid nodules. The fluorescence polarization images provided quantitative contrast at the cellular level for thyroid cancer detection. The results showed that cellular level fluorescence polarization values are not specific to each patient. The research team of this project included Dr. Anna Yaroslavsky, Director of the Advanced Biophotonics Lab, Dr. Andrew Fisher and Sarah Pantanod of UMass Medical Center, Dr. Leah Joseph and Thomas Ackerley of Lowell General Hospital, and Alana Musikansky of the Massachusetts General Hospital Biostatistics Center. The project was funded by the UMass Center for Clinical and Translational Science Medical Device Innovations Program, the Massachusetts Technology Transfer Center ACORN Innovation Fund, and the NSF I-Core Teams Program.